Hi students, my name is Said Saifullah and you're watching Mr. Lecture. So today we are going to study the topic cell surface membrane. So first of all, let's understand the structure of cell surface membrane. So the cell surface membranes are made up of phospholipid layers. So what are phospholipids? Phospholipids are a combined structure of a phosphate group and three fatty acid tails. So the phosphate group is hydrophilic, means it's polar, and the fatty acid tails are hydrophobic, means they are non-polar. So the structure of phospholipid is somewhat like this. So this is the phosphate head, and these are the fatty acid tails. So the cell surface membrane is made up of such kind of combined layers. So when the phospholipid layers are mixed with water, they form either of two structures. One is micelles and second is bilayer. Micelle means the phospholipid layers make a ball-like structure, and the bilayer, it's two layers. One is outer layer and one is inner layer. In 1972, there were two scientists, Singer and Nicolson. They both put forward a hypothesis for the structure of cell surface membrane, which they termed as the fluid mosaic model of cell surface membrane. So let's understand the fluid mosaic model of cell surface membrane. So this is the diagram of fluid mosaic model of cell surface membrane. So let's understand this diagram. First of all, let's see phospholipids. So the membrane is a double layered membrane in which the phospholipid tails face inwards, pointing towards each other and forming a hydrophobic interior inside. And the phospholipid heads face outwards into the aqueous environment that surrounds the cell surface membrane. There are three factors that affect the fluidity of the membrane. First of all, the saturation. If the fatty acid tails are unsaturated, means they have a double carbon bond in them. So the membrane will be more fluid, and otherwise it will be less fluid. And the second factor is tail's length. If the tail is long, so the membrane would be less fluid, otherwise it would be more fluid. And the third factor is temperature. So if the temperature is decreased, the membrane would get less fluid, otherwise it would get more fluid. Now we are done with the details of phospholipids. So now let's come to the proteins. The proteins in the cell surface membrane are of three types. They may be on the outside layer of the membrane, or maybe in the inside layer of the membrane, or maybe spanning the whole membrane. So in that term, they are called as transport proteins. And there are some carbohydrate chains attached with the proteins on their surfaces. And this combination is called as glycoprotein. When the carbohydrate is combined with the protein, so this combination is called glycoprotein. And maybe there are some lipid molecules and the carbohydrate chain is attached with them too. So this combination is called glycolipids. So the glycolipids and glycoproteins are very important in the cell surface membrane. So now let's see the functions of the glycolipids and glycoproteins. The first function is receptor molecules. The glycolipids and glycoproteins act as receptor molecules, means they bind with specific substances at the cell surface membrane and detect them. The second function is cell-to-cell recognition. Some proteins, glycolipids and glycoproteins, act as cell markers or antigens, allowing cells to recognize the things they attach with. And this is done with the help of the carbohydrate chains. Third function is transport proteins. The proteins, such as the ones which are spanning the whole membrane, act as transport proteins. So their main role is to provide hydrophilic channels for the ions and polar molecules which cannot enter through the phospholipids to enter the cell surface membrane and have a smooth exchange of materials. The fourth function is enzymes. Proteins act as enzymes. The example of this is the digestive enzymes in the cell surface membrane of the cells lining the small intestine. So the proteins, they act as enzymes to digest the food particles. Now last but not the least, the function of cholesterol in the cell surface membrane. So cholesterol is found in the cell surface membrane. So like the phospholipids, the cholesterol have a hydrophilic tail, a hydrophobic tail and a hydrophilic head. So the cholesterol fits into the phospholipid molecules with their heads at the cell surface membrane. Cholesterol is important in the mechanical stability of the cell surface membrane. This is done by the cholesterol when it fits into the phospholipid molecules and decreases the fluidity of the cell surface membrane, so hence stabilizing the cell surface membrane. Secondly, the cholesterol prevents the ions or polar molecules to enter the cell surface membrane. Now you may say that this is done by the phospholipids too, so it's not important. But it's important in some rare cases, such as in the myelin sheath which surrounds the nerve cells. There, the cholesterol plays important role in saving the leakage of nerve impulses from the nerve cells. So that's why it's important for preventing the ions or polar molecules to enter the cell surface membrane. So this was all about the fluid mosaic model of cell surface membrane. If you found this video informative, so please like and subscribe to our channel for more such informative videos. Thank you very much.